I feel like I'm running a true facts video sometimes. So today we're going to talk a little bit about variability, environments with a lot of variability. And I'll admit to you that this is not my favorite way to organize the information. But we've already talked about aquatic environments, we've talked about terrestrial environments, and now the main differences between those being, the variability of temperature, how much water you have access, but there can be even more variability here. So one type of variability we're going to talk about here in the first one is how systems vary in time and in space. So in terms of time, we can have day-to-day changes in our weather, or we can have year-long, century-long changes in things like climate. So the difference between weather and climate, weather tells you whether you need a jacket today or whether you need an umbrella today. Climate is going to tell you when is a good time to book a ski vacation in Colorado. So climate is averaged over years. You probably wouldn't go on a ski vacation in Colorado in August. That's its overall climate. But if you're on a mountaintop, sure as hell, there could be a little bit of snow, a flurry any day, and that's weather. Some events are huge, but have a big impact, things like tsunamis and hurricanes. Other types of variability, you can encounter things like forest fires. Some systems have evolved to utilize forest fires. So the more extreme the events, the less frequently they tend anyway to occur, although a lot of things are changing right now. So large scale variability and small scale variability can also happen in spatial scales. So you're talking about what type of land do you have, what soil type do you have. They can happen on large or small scales. Some people, my dad lives in Seminole, and he's constantly bitching that we get all the rain in St. Pete and it never makes it to Seminole, right? And that's pretty small scale. Good traffic, I can be over there in 10, 15 minutes. Yet there is variability on the small scale and also on a large scale area. So the faster you move through space, so if you go through space, the smaller the scale of your spatial variation, the shorter the temporal scale. So it depends on your size, what scale of spatial variation is important to you. So if you are a cockroach, your large scale variability might be somebody's house, but small scale would be running under the fridge or under the stove. If you are a deer, then small scale variability might be the plants that you're standing next to, but large scale could be miles and miles and miles of forest that you are going to move through. So this is giving you an idea, again, of this, the extent of the effect is usually related to the event's duration and time. So things like storm fronts and thunderstorms, if you look here, the x-axis is spatial dimension, the y-axis is duration. So we have coastal fronts, they last, a year, thunderstorms last on a day, and they tend to have scale variability more in the 1 to 10 kilometers. Whereas if you get out here towards hurricanes, they're pretty huge. They can start having a huge kilometers of impact and variability as they move along. Variations in topography and geology do occur, but fortunately they tend to happen at a much, much slower pace. So for example, the formation of the Hawaiian Islands is a variability in topography and geology, but it takes a really, really, really long time to make a new Hawaiian island. It can happen, but when you get short scale variability in geology, it tends to be something quite spectacular, like a volcano that blows up half an island or part of a mountainside. Those are all things that happen in a geological sense, but fortunately, most geological events happen over small scales. Again, when Krakatau was blown off the planet, it's kind of a big deal. So when you have a lot of this variability going on, you will often see the evolution of phenotypic plasticity. We've already talked about this a little bit before, because the traits you express to the environment are your phenotypes, and they result from your genes and the interaction of your gene with the environment. So it has a genetic basis, but depending on what your body needs, you're going to express different genes. We've already talked about this when we talked about temperature variability, because we know organisms can produce more than one enzyme depending on the time of year. And those enzymes will have slightly different bonds, and those bonds enable it to work well in warm temperatures or in cold temperatures. And so, as it says here on the bottom, there is kind of a trade-off, because a phenotype that is great in one environment might not be suited to the other. And so this is showing you this idea of phenotypic trade-off, a situation where one phenotype has a better fitness in terms of passing on genes. My bird is just not into these morning lectures. Whereas the other one is fit in a different type of an environment. Up until this point, we've talked about phenotypic plasticity using examples of genes that can change in an individual's lifetime. So the acetylcholinesterase that we've been talking about, that enzyme, you can make one copy in the winter and one copy in the summer. But in this chapter, we're going to talk about some of that plasticity that doesn't change. It happens during development. And once that choice is made, you can't undo it. There is no, control X, oops, let's revert to our previous state. That can't be done. And this happens in some things like Daphnia and so some frog species that we're going to see. And so there is a choice in terms of long-term fitness for that organism. They're going to have to pick which phenotype to express in the environment, and then they're stuck with it for their life. So this is an example of a gray tree frog. And during their development, when they're little eggs, if there are chemicals in the water that indicate there are a lot of predators, they will develop a body type that allows for fast escape. But if there aren't any of these chemicals that are known as cheiromones, K-A-I-R-O-M-O-N-E-S, I'm a terrible speller, but yeah, it's cheiromones. If there aren't cheiromones around, then they will be a sleeker, they will grow faster. So what's the advantage of each? Well, we got this little guy here on the bottom, like more purple frog, very, very burly and has a great burst response, but it will take longer to reach maturity and be able to reproduce, because it put a lot of energy early on into having this bigger tail, enabling to escape predators. Because if it doesn't escape the predator, it's never going to live long enough to have babies anyway, so it doesn't matter. Whereas the other organism, the other type, the phenotype that it could express, allows it to grow up to be an adult faster and start reproducing. So it will probably have more reproductive events during its lifetime, provided that it gets there and doesn't get eaten first. So this is one example of phenotypic plasticity, a change that happens during the development. It can't be swapped back once the organism is become an adult. So what are some of the environmental clues to alter its phenotype along the way? Well, hopefully it's using cues that are useful. So one of them are the chemical cues in the water, the cheiromones during development. That can be something that will cause them to change their development. Another thing you can see is to detect the presence of competitors. They may not matter if resources are abundant, but there are cases if there are a lot of other tadpoles around, you'll see the development of the tadpole change again. And then also, how much food can you get from your environment? Is there enough food for you to support growth and then reproduction? So that's the example of, if you will, the directional phenotypic plasticity. You can't go back, but there definitely are these examples that we've already talked about before, like acclimation. So the environment can, you go to the top of a mountain, so you go to Colorado to go skiing, right? There's less oxygen up there. And so your body is going to have to respond to enable you to continue to function. So you go up there, your body chemistry changes after a week or so, but when you come home, your body chemistry will change again to adapt to having more oxygen around. Things that happen rapidly tend to be more reversible. I mentioned before, getting a tan at the beach is technically a form of phenotypic plasticity. I'm not, like I said, maybe it protects the skin, things like that, but that can be undone. So things that change in your lifetime can tend to happen more quickly, whereas some of these slow responses are irreversible. And so if you're living in an environment where there's lots of rapid fluctuations, you're going to want to be able to respond rapidly. If you're in a more stable environment, slower changes are probably going to be what's preferred. So what are some of these adaptations to variability in the environment? Well, we're going to talk about adaptations to enemies, adaptations to competitors, and adaptations to mates. So this is another example of species altering their growth and body shape in response to predators. And these allow these guys to, so these poor little small euploides can be smaller, or they can have a larger winged version. And once again, there is a predator around that gives off chemical cues, the pheromones. So it's not like a pheromone where you intend it to give information. It's something that just kind of sits there. So you can think about these chemical cues like the smell of popcorn. It's certainly there, and everybody knows somebody just made popcorn, but it wasn't given off intentionally. It's just a side effect. And so it's like the smell of garlic. These are things that are just around. They're not necessarily direct chemical signals from one to the other. However, this little euploides still does some eavesdropping, and when it senses those chemicals, it grows a little bit faster, so it can't fit into the mouth of the predators. But now the predators are like, hey man, I need to eat something, so they can also respond by growing larger. And they can eat a bunch of the large wings, euploides that are hanging around. However, eventually they eat all the big guys and they're slow, so they can't get a hold of the little ones. So you end up in this evolutionary arms race alternating between larger and smaller organisms. And these are just adaptations in the predator-prey cycle. The prey wants to live long enough to reproduce. Their day is going to be completely ruined if they're eaten and killed by their predator. And the predator needs to have lunch so it can continue on with its environment as well. So plants get in on this game as well. This is looking at the Virginia pepperweed, and it is often preyed upon by herbivores. A lot of times when I talk about herbivores, I mention things like cows, right, or deer along the way. But in fact, many of the herbivores on the planet are really teeny tiny. I think whatever's been eating all of my flippin' peppers are actually little caterpillars. I already had some butterfly caterpillars absolutely decimate my parsley this year. So insects are super important. And here we got a little aphid hanging out on the right-hand side. So we're looking at this pepperweed, and we're looking at the glucosinolate. I can never say that. It is a compound, a chemical that makes the leaves difficult to consume. And the idea is that these leaves will then attract fewer predators. And then you look at the number of trichomes per leaf, which also makes it harder to eat. So you can see that you have higher levels of the chemical compound in the plants that have been induced. We also see more trichomes per leaf. And as you go over the control ones that have the lower levels of chemicals and lower numbers of trichomes end up with more herbivores on them. So sometimes getting bitten is a physical cue to the plant to then respond, that change its leaves and change the amount of chemical compounds to help make them difficult to consume. Why don't they just do it all the time? Why aren't they just ready? Why aren't they prepped and ready to go? Because remember, everything costs money. They're going to have more energy to make these chemical and physical defenses. And that's energy that can't be put into seeds for reproduction. So being able to wait to see if you're going to be attacked is an advantage, and then you just respond to it, saving energy for the time that those guys aren't around. There's also a competition for resources. Organisms can change their metabolism and physical structure based on high and low competition, or whatever type of food is around. They can spend more time looking for food depending if they are an endotherm or an ectotherm. And this is an example of a Burmese python. So what's really interesting about pythons, so they're really big and they are ectotherms, they're cold-blooded organisms. but they only need to eat like maybe once a month, maybe 3 times a month. That would be it. But a lot of times it's only once a month. So what they do in between meals is they actually catabolize, they break down their own intestines. They don't want to have to carry around the extra weight, and they don't want to have to waste metabolic energy maintaining their intestines. So when they start to go into a period of fasting, they actually break down. So if you look over here on the right-hand side, you see the intestines of a snake that were fasted, and they're absolutely teeny, teeny, tiny. As soon as they eat something, the messenger RNA goes through the roof and quadruples the intestine. They double the heart, quadruple the heart size and double the length of its intestines. Look here, the next one is just 2 days post-feeding. How much bigger those intestines are. So it actually has phenotypic plasticity in its intestines and the size of its heart. That's something that in humans we can't do. Like, we would totally go to the hospital for that. But they have this in order to respond to different levels of food in the environment. So if they're in a fasted state, they don't need all of that metabolic energy to be wasted, if you will. So once it eats, it jumpstarts it. It's got like a pop-up tent of intestines, quickly utilizes all of those nutrients, and then goes back to a period of fasting again. And this is really pretty kind of an extreme example. Now, when you are in the environment, sometimes it's harder to find a mate than other times. And so we see differences in reproductive systems depending on what an organism can utilize. If like in the case of snails, or I'm trying to think of, well, actually snails do this a lot because the gastropods, nudibranchs do it. If males or females are in short supply, you will often see hermaphrodites. So these are going to be individuals that produce both male and female gametes. Some hermaphrodites are sequential, meaning they have their girl bits and their boy bits at the same time. They have all the plumbing just sitting there. Sometimes hermaphrodites are sequential, so they are male and turn female, like we see in the anemone fish that you saw in Finding Nemo. Or you can have something like a grouper that is female and then becomes male. And can occasionally, there's some little blennies out there that can switch back and forth between male and female depending on what they need, which is nature just always finds a way. So it costs a lot of money to do this. However, again, metabolic money. However, this guarantees that the organisms will have higher reproductive success because they will have enough male or female gametes around depending on what they need. Now, even when you have organisms that are simultaneous hermaphrodites, so they have both male and female sexual organs on the same organism, you don't usually get selfing, where they will actually fertilize their own eggs, because that's going to decrease the fitness. They are not going to get as much variability, and you can end up with inbreeding depression. So you don't have new genes coming into the system to make up for to replace some of the bad copies of genes that might be there. However, if that's the only opportunity for reproduction an organism has, then they're going to go for it. So this is a snail, a common pond snail, and they will delay mating if mates are unavailable. But if they're not, they will wait, and then they can actually fertilize their own eggs if they need to. So these have been adaptations to enemies, competitors, and mates, but they can also clearly evolve adaptations to abiotic conditions. This is showing us adaptations to temperature. We have talked about this in the past, so we know these guys actually produce different isozymes that allow the goldfish to swim fast in lower temperatures. And then as it gets warmer, they can swim fast in higher temperatures. Actually, I think the example we looked at before was a trout, I think it was. But the same thing happens here in the goldfish. So what happens if you have a well-functioning version of acetylcholinesterase? Well, it's an enzyme that is involved in muscle contraction. And so for our little friend, the goldfish here, they can swim faster. So on the y-axis here, instead of having enzyme activity, you actually have speed in centimeters per second, but the graphs look super similar. So the speed in centimeters per second there's two lines, one between 0 and 25 with a peak about 15, and then the second one begins about 12 and having a peak about 25 degrees. These different versions of the enzymes are produced depending on what environment they're in, and it has a direct benefit to their swimming speed. If you were the trout, that means you can swim faster to catch prey like a goldfish. For the goldfish, they're really not known for having any superpowers or eating other fish, right? They're usually pets and stuff like that. This means that they can get away. Although they are super invasive, we will talk about how horrifically invasive the rather low key and modest goldfish can be. Now, other adaptations to temperature variability can actually be behavioral. And in this case, it is just exploiting a micro habitat. So that's just a little spot inside a larger area that has very, very different conditions. So the example they're showing you here is a lizard in a burrow out in the desert. Our friend the kangaroo rat does that as well. They live in a desert. It's super, super hot. So they can go down into these burrows. It doesn't take much, just 2 or 3 feet, to really drop the temperature down by several degrees Celsius. So they can just cool off. So they're exploiting a microhabitat if it gets too hot. If they get too cold, then they have this opportunity to go bask on the rocks. So they actually can very effectively regulate their body temperature, even though they're technically a cold-blooded organism, but they're doing it by moving. And we'll see a paper in CompPhys where they maintain it between 30 and 37 degrees Celsius. Super, super great regulation. But microhabitats are very important in terms of surviving in variable environments. And sometimes it's the only place that organisms have to live. Whole groups of mice rely on the area just underneath the snowfall. It's super, super cold on the top of the snow, but the snow is a little bit insulating. So they actually live their entire winter in this tiny micro habitat between the end of the snow and the soil to help keep them a little bit warm. You can also adapt to water variability. We see this a lot in plants because they can't move. They're going to be their adaptations will reflect long-term climate. So things we have mentioned before, you can close your stomata if you have the right kinds of photosynthesis, or you can even do it for a short period of time. Or you can adjust how much energy you're going to put into your roots as opposed to your leaves. And this shows us an example of 3 types of plants: gray hair grass, mouse-eared hawkweed. Does it look like a mouse? I don't think that looks like a mouse, right? But it doesn't matter. I didn't get to name it. So 3 kinds of plants. The blue is when we have abundant water, and the pinkish color is when there's scarce water. What you'll see is in all 3 species of plants, the root to shoot ratio is lower for the abundant water than the higher water. So if there isn't a lot going on, if there isn't a lot of rain, organisms often put more energy into their roots rather than their leaves. The roots are one way that plants can, if you will, make a comeback. If the conditions are really bad, they put more energy into the roots, they can get more moisture if it's there, and they can overwinter or survive the drought if that's what they need to do. So this is something that plants can do in a short scale to adapt to variability in water. There are also things organisms can do to adapt to salinity. So one adaptation to increase or lower levels of salinity is to change the amount of organic solutes that are in your blood. So we don't really do that too much. We are high-end regulators, but a lot of things like this little copepod here on the bottom, they have an open circulatory system. They're a little bit more adaptable to change. So if things get too salty, what they start to do is dump amino acids into its bloodstream. It is pretty costly. However, having more bits of junk dissolved in its body helps it to keep water inside. Otherwise, it would just rush out into the salty tide pool. And one little copepod's body is not going to dilute that water enough. So organisms can change the osmotic potential of their body fluids by adding different organic solutes to it. And this is what we saw in sharks as well, because they have urea to help them with their osmotic pressure, but then they use TMAO to counteract that. So urea, amino acids, some organisms use glucose. These are all little bits of junk that they will dissolve into their blood to help them keep water inside rather than having it leak to the salty environment around it. It might seem like it's a waste of time to make a bunch of amino acids. Why don't they just keep more salt or something like that inside their body? Well, the problem is things like sodium and chloride are super important for your heart function and your brain function. So it's dangerous to use salt ions as an osmolate, one of these organic solutes. It's much, much safer to use something more benign like an amino acid. Now, there are, of course, adaptations to oxygen. We can change if you're going up and down, say you're a bear, not a human. They go up and down as well. They can change their breathing rate. You can change the number of red blood cells in your body, and you can also increase the heart rate. So this is something that you see on a short scale variability rate, but you also see long-term evolutionary adaptations in populations that always live at elevation. So if you look at the native populations that serve as Sherpas on Mount Everest, they have a evolutionary adaptation to being able to breathe very low oxygen levels. And we see this in the Himalayan geese as well. They have different kinds of hemoglobin. They have changes that they are born with. No matter how long you or I went to go live in Nepal, our bodies will never be as good at getting oxygen out of the air as those populations are. So you can have adaptations that are short-term that would be in the realm of plasticity within your lifetime, but then there's also adjustments that can be made on an evolutionary time scale as well. So other ways you can get around variability in the environment are just to, like, well, move, right? We mentioned it a little bit in microhabitats, but what if you just want to be a snowbird? You live someplace when it's nice and cool, you live up north. When it changes, you come on down to the little latitudes to get some warm weather. So those are adaptations as well. And we see migration in a large number of organisms, everything from the lowly monarch butterfly. We see migration in hummingbirds. We've seen, you see migration in Africa, water buffalo. So they move over large expanses from one region to another for nutrition, for mating grounds, lots of reasons to move across the country. The decision to migrate is a plastic behavior in response to environmental conditions. So a lot of times the change that initiates this migration would be temperature, but it also can be photoperiod. So photoperiod refers to how long each day is, how much daylight do you have. And so here the example is monarch butterflies going to wintering areas in southern North America, down by where we are. Some organisms do stay behind. Not all organisms continue their migration patterns. It's one of the bits of variability that you see in evolution, just in case, you never know. And animals that migrate tend to have a very efficient mode of transportation. If it costs too much energy to get somewhere, you might starve along the way. So it's not unusual that many organisms that migrate get there by flying. Flying is a super cheap way to get around once you're off the ground. So you can see hummingbirds, terns, butterflies undergoing these kinds of migrations. Fortunately, not gators and not crocodiles. Can you imagine a mass migration of gators like from Florida up to Lake Michigan every year? That would, oh my gosh, that would be so crazy. Other thing organisms can do to adapt to this is just store up food. So you can have little pikas. They look like mini hamsters that live in the Arctic. They store up food during the summer. They put them in a little hay pile there so that they have food over the winter. And plants can do this too. They can store up reserves in their roots so that they will actually have energy to overwinter and come back. Again, if it's going to be harsh, plants will often put energy and storage into those roots. The surface of the plant may die, but then it will come back the next year. So this is actually coming from the roots, not just some seeds that were hanging around. But storage is another way organisms can adapt to these environments and make sure they have the energy to make it through. Even more fun ways to adapt to harsh weather is going to be to use dormancy. So this is when organisms actually change their metabolic processes to adapt to what's going on outside. So one is called diapause, and it involves the partial or nearly complete physiological shutdown in response to unfavorable conditions. And diapause is very common in insects. It often corresponds with the production of some antifreeze compounds in their bodily fluids so that they don't actually freeze. If your blood were to actually freeze, the expansion of the water would then cause poking through the membranes and could actually cause death. So they have antifreeze compounds, and then as they mentioned here, they will often even dehydrate themselves even further. So that way they don't have to worry about they're already dehydrated, so when the water freezes, it's not as big of a deal. The next one we're very familiar with is hibernation. So individuals will reduce their energetic costs by lowering the heart rate, decreasing body temperature, and we see this a lot in mammals, particularly small mammals like ground squirrels. And their energetic costs change, that they wake up at different times depending on what reproductive success they'll need. But this is looking at a little winter chipmunk. He's all curled up, he looks super cute. And so they'll slow their heart rate, and they can get their body temperature close to 0 Celsius. So not quite freezing. It only takes a couple of degrees difference to keep everything moving around. Then another one that we see that's very short term is known as torpor. So torpor is like a mini hibernation. It occurs basically overnight. So it's a period of dormancy that reduces the body temperature, but it happens very rapidly. It's not something that happens for months, it just happens overnight. And so this is looking at the West Indian hummingbird. It loses much of its heat to cold temperatures. Anything that's going to be small has a really high surface area to its body mass, and it's going to be much smaller organisms, whether it's a hummingbird versus a bald eagle, or it's a human, like a teeny human as opposed to a larger human. Smaller organisms will lose heat more quickly. And if you're a small hummingbird, it makes it even harder. So you're going to run out of food and energy overnight, particularly on days when it's cold. So what do? Well, you can see here that the torpid animal actually drops its oxygen consumption down. If it was an active hummingbird, its oxygen consumption would be, I'm sure it's grams of oxygen per hour, here about 13. In a state of torpor, it drops that down to about 6 or 7. So you're talking cutting its metabolic rate in half. And it leaves them in some unenviable positions, which we're going to see here in just a second. And then the last type of dormancy you will see is called estivation. And this is basically hibernation when it's hot. So you shut down your metabolic processes during the summer in response to hot or dry conditions. And we see this in desert frogs. You might think that an animal dependent on water wouldn't want to live in a desert, but nature finds a way. So there are actually types of frogs and lungfish that will burrow down. They make a really yummy, tasty mucus cocoon, and they live inside that until the rains come back. And you're like, I don't want to live inside a mucus cocoon. Well, that's a good thing because you don't have to, right? But you see this in so the spadefoot toads, you can see it in lungfish, but also snails, desert tortoises, and crocodiles can use estivation when it's just too hot outside. Now, in terms of freezing, again, I'd mentioned it lately before, but these organisms can actually produce antifreeze chemicals. They work in a couple of different ways. They can work to prevent ice crystals from forming, or they can work to trap ice crystals so once they form, they don't grow. Some organisms will burrow to temperatures. There are a few amphibian species that can freeze nearly solid and it doesn't seem to bug them too much. But here it says they have their antifreeze compound, so that keeps the crystals between the cells rather than inside of them. And it's really kind of unique. Each animal who allows itself to freeze or has to survive in sub-freezing temperatures produces a different type of compound depending on what it needs. Freezing is a much bigger deal on land. If you're in the ocean or in the water, remember there's a higher heat capacity. And if you're in a freshwater lake, it's usually pretty unusual that that water is going to get below 0 Celsius when your body parts would start to freeze, because you get that nice layer of ice on the top. In the ocean, it's different because the freezing point of seawater is actually about -2 Celsius. So that is below the freezing point for the water that would be inside a fish. And so they will actually have their own antifreeze proteins. So let's take a little break here. I'm going to go for 2 minutes, or 3 minutes, go grab something. We're almost done. I'm going to show you the hummingbirds, and then we're going to talk about optimal foraging theory. So go grab a drink real fast, and we'll be right back, and we'll watch some cute hummingbird videos. Because you got to see them in torpor. It's adorable. hear this really peaceful breeze coming through someone's microphone. It's really like, oh, it's like the wind out on the beach. I mean, we're not there. I just have my fake picture. So, all right. So we're back and we want to see some cute hummingbirds. I know you do. So remember I mentioned torpor. Was that Bregan?
Oh, I'm sorry. I couldn't hear you. I got it.
Okay. Okay. All right. Good. Good. No. I think we all know that every day you wake up to a new version of Zoom.
Oh,
right?
It's
It could be my microphone if I tripped over it or something. I didn't. When all of this went down, I didn't have like this huge tech studio. And my husband's music studio is designed to like record saxophones, not do Zoom meetings. So we're getting by. All right. So we mentioned torpor. And it's really hard to describe unless you see it. So here's going to be some adorable little hummingbirds. So this is looking at 10:30 p.m. A hummingbird has fallen asleep on the feeder. So it actually hangs upside down. It's kind of embarrassing. They actually have little extra joints in their feet so that they don't fall. Let me stop this real fast just so I can get the sound. I don't remember if it makes sound, but if it does, it's the cutest little peeps you've ever heard. Okay. So here he is. And the other's like, dude, someone had too much last night. He's still just sitting there. So torpor is a pretty deep state. You can almost think of it as like he doesn't notice the deer. He doesn't notice anything. He's just hanging out there. So what happens to them when they do this? Well, this is, oh no, stop, it's cute. This is actually a metabolic state. And to show you what happens metabolically, we're gonna go to one of my favorite video sources, which is TrueFacts. And they're gonna show you with an IR camera what happens to them when they go into torpor.
That's a moth. So anyway, they're out there flip-flapping all over the place, hitting the juice with their freaky tongue, and they're tweaked out. You've been there. It's like cramming for a test with a 2-liter of Dr. Pepper. All jittery and you have to pee every minute. I'm telling you, everything's up to 11 with these tiny puffball junkie birds. And when they're hopped up looking for that next flower fix, their little wing parts are going at 30 to 40 flaps per second. And their heart, forget about it. Well, not before I say it. It's like a speed course set at an EDM concert. Somewhere between 500 and 1,000 beats per minute. Now forget about it. And they run hot. You can see it on these tie-dye cam. Friggin' science hippies. 104 degrees Fahrenheit, which is approximately, look it up on Google yourself, Celsius. Friggin' metric system. Takes all the romance out of measurement. I want my units based on the size of a king's unit. That's what a shitload is. 1 king's loaf. Anyway, these jacked up hummers are caught in this crazy trade-off. If you want to take 1,000 drinks, you have to be quick in all directions. So you get...
All right, so that's showing you the high body temperature, and there are the really active ones are red, and they can actually get down to the blue range. So they can produce that much less heat. It's not just oxygen, but actual heat that reduces their metabolism. And that is a really funny video. If you have an extra 12 minutes today, you're not going to be disappointed. I did not even realize how the hummingbird beaks worked, but apparently they're little pumps. Who knew, right? Yeah, very funny. It's got to be nice. You have such a great YouTube channel that scientists are sending you their footage to make their animal famous. It's like, oh no, I want to do that. I want to do that. There's one on these like predatory tidal surfing snails. I think, Todd, didn't we watch some of that in Marine? If I didn't, we totally missed it. Yeah, and you're like surfing snails? Who? I didn't even know about that, but scientists know about TrueFacts. And they're like, I got this footage. I want to do it. I want to do it. So, all right. So let's go.
Watch the one about the giraffes.
Oh yeah, giraffes. Giraffes are funny. Actually, a really good one to watch is on the anglerfish. He has 2 of them now. But talk about weird reproductive strategies. Your book didn't mention it, but anglerfish in the deep ocean may never find each other. So when the male finds a female, he attaches to the female, dissolves his head, and just becomes a little sperm sac and travels around with the female. That's it. That's their end game. But there's so few females around that's the strategy. You find a female and you just stay no matter what. I mean, nature has found a way to make anything happen, and that's another funny. I think he has 2 on them now, because people got jealous of the video footage. Okay, let's finish this up. So we finished the chapter up by looking at variability in food quality and quantity. I think this, wait, no. If I don't turn off the video thing, sometimes you can't read the screen. Learning things the hard way. So getting food often involves foraging. And foraging is a behavior that changes depending on which organisms are in the environment. So because your resources are going to vary in space and time, one spot that had a lot of food might not have a lot of food anymore. Maybe it ran out. There isn't really going to be any single foraging strategy that's going to maximize your organism's fitness. And so they have a lot of plasticity in how they look for their food. And they generally choose a strategy based on a couple of different ideas. How far is it from where their nest is? So that's central place foraging. If you're an organism that has a nest or a burrow, central place foraging will be important. There's another one that's called risk sensitive foraging. And this is something important for things like fish, where you might be looking for worms, but there's big predators around. There's optimal composition and diet mixing, and this has to do with choosing the proper type. If you're a bird that likes to eat insects, but you're not picky about whether it's a cricket or a beetle, you can look for the optimal composition or having just the right amount of each one in your diet, and you can mix it up. So this is looking at the idea of central place foraging. And the further away it has to travel from the nest, the more energy it's going to burn looking for food, and the more time it's going to spend searching for food. So there is a interplay between the number of prey caught and the amount of time they will spend traveling and searching. The exact units is going to differ for each species, but there is a point where this curve increases sharply, but then starts to level out to an asymptote here. And organisms tend to try to stay right here where the slope is really, really steep. So for the time you're putting in, you're getting an awful lot of prey that are being caught, because otherwise you have diminishing returns. As the individual furthers farther and farther away, it takes more energy, so there isn't as much left over when they come back. This is particularly important if they're feeding young birds, because the adult may not even get to eat any of that because it's feeding all those little birds that are yelling at it. Like my bird that wants some millet. And I'm like, dude, just wait, right? So this is looking at how they make these decisions. You have the optimal number of prey caught, the optimal search time, and you want that where it's steeper and where you can actually before it starts flattening out. So if you're going to pick a site that is very far away, it better have some really good food that you can bring back. Risk sensitive foraging is changes in behavior based on whether you're going to get eaten or not. And this is an experiment that looks at creek chubs with predatory fish. And basically, the take-home message is if there's a lot of food, it just might be worth it. So on the top, we have a graph showing the fish in a refuge. So they actually had like a safe zone. And then they dump these bloodworms out there to try to lure the fish over near the predator. And not surprisingly, if the fish had to choose between one predator and 2 predators, they're like, well, that's a no-brainer. Let's go to the left-hand side. Let's just go where there's one predator. However, if you put enough food, as you move down more and more and more, they're like, well, what if we give you 33 worms instead of 17? Nope, no thanks, not gonna do it. But if you made it a lot more attractive, say 0.5 worms instead of 0.17 worms, now it was worth it. There's enough food that fish feel that they can jump over, grab some worms and get back before the predator can get them. So this is, overall, they're going to look to avoid predators, but since there's probably never going to be a time where there's no predators, they're going to pick the area where they get the best food return. If it's 0.7 worms, it's not worth it, but if you can double that, then it might be worth it. Double or more, right? The other thing that comes into play is how long it takes you to open up your food, right? The handling time. So if you're choosing something to eat, you're going to want to be able to open it. And I think we've all been there where you have that bag of Doritos, potato chips, or maybe worse, a bag of carrots or broccoli. Like, why do they make the vegetable bag so flippin' hard to open? It's supposed to be good for us, and now you're making it hard to open. So you're sitting there like spending all this time or trying to open a peanut butter jar, right? And that's handling time. The amount of time it takes a predator to consume a captured prey. And so the predators are going to look to eat species that give them a lot of energy back for the amount of time they spend handling it. This is something that you see often in birds that are feeding on shellfish. So smaller shellfish don't have a lot of energy in them, and so they're not as beneficial, but taking something really, really large, like a really big clam, might take forever to open. So by the time they could have had 10 medium-sized clams and the time it took them to open that really large clam. So organisms will look to optimize their handling time as well. Or like, hey, that's a really tasty worm and it doesn't have any hard bits. I think I'll just eat the worms, right? And that's what you see here with diet mixing. Some foragers will consume a varied diet because they have different handling times and they might not have all the nutrients they need in one specific organism. We do that quite often. And this is looking not just humans eating different things, like eat your vegetables, your mom probably told you that. These are the nymphs of grasshopper. And so they mix the type of plants that they eat. If they're given a choice, they will mix between mesquite and mulberry. It's not that they couldn't just eat mesquite or couldn't just eat mulberry, but when given a choice, they will mix the diets because they will get better nutrients out of it. And you'll see diet mixing when one species runs out. So if someone was eating crickets and all the crickets are gone, they might switch over to beetles because they have to. And then the beetles start to go down, but the crickets have made a comeback. So you can see prey switching in there as well. So now we're going to go into the analyzing ecology next. That's going to be our correlation section here. This is very, very important because you see a lot of correlational studies out there. And just because two variables look like they have a relationship to each other, it doesn't mean that they do. And this is one of my favorite. This is looking at global average temperature in relationship to the number of pirates. And you can clearly see that global warming is being caused by the fact that we don't have pirates anymore. Back when there were lots and lots of pirates, the temperature was lower, but now there's only 17 pirates or so in 2000, right? And now it's warmer. Clearly, you have to start having people go back to piracy. Okay, that's not really the case. But there are correlations between SAT scores and volcanic eruptions. There are correlations between drowning death and ice cream. Ice cream does not cause people to drown. Okay, it does not cause people to drown, but the correlation is there. So your book went into pretty big depth about the what a correlation is. So for those of you who want to do analyzing ecology on your own, now is a good time for you to pop out. Those of you who want to stay, give me a second and I'm going to log in. I won't make you watch me do that though. All right, so here we go. We have question number 1 from Analyzing Ecology, and I'm sure it's frustrating for you too when it doesn't fit in the screen, right? Let me go ahead and pull up the screen, get the audio going. Shouldn't be too big of a video, but
Correlation is a tool used to analyze data. With correlation, we analyze how one variable changes relative to another variable. Let's take a closer look at how correlation is used in scientific experiments. In a study of hummingbirds, scientists hypothesized that oxygen consumption is affected by air temperature. Looking at the graph, there appears to be a relationship between temperature and oxygen consumption. As temperature increases, oxygen consumption decreases in a linear fashion. We can perform several statistical tests to learn more about this relationship. With correlation analysis, we describe the type of relationship and the strength of the relationship between two variables. We can also calculate a trend line that best fits the data with a method called regression analysis. The trend line also gives us a visual representation of the correlation between the two variables.
no, that's cute. I didn't see the hummingbird fly on my little screen yesterday. Okay, so let's go over here to our first question. Number 1, what is correlational analysis? Okay, what do we got on here? A method for determining the average value of a data set. No, we're not doing that.
I think your screen is blurry.
It's what? It's blur. Okay, thank you.
Impossible
I see the chat just exploded. That's because it'll show you the video and then it doesn't show you the text. So the text looks like crap, but you got to see the little hummingbird. So, you know, let's go for the text. Let's go for the text. I think that's probably more what you want to see. Okay, so turn off. Better? Yes, okay. First question is, what is correlational analysis? We have a couple of different selections here. Statistical tool used to calculate the best fit line. We don't calculating any line. That's actually something you do in regression. Statistical tool to measure the strength and type of relationship between two variables. A method for determining the average value of a data set, or a method for determining the independent variable and the dependent variable. It can help determine the independent and dependent, but that's generally something that you do. And because in this method, we're not making any kinds of calculations, we're just plotting it on a graph. So the answer for this one is a tool to look at, to measure the strength and type of relationship between the two. Okay.
Correlation analysis describes the type and the strength of the relationship between two variables. We can describe correlations in several ways. The correlation between two variables can be positive or negative. In addition, the correlation can be described as linear or curvilinear. A positive correlation occurs when, as one variable increases, the other variable also increases. A negative correlation occurs when, as one variable increases, the other variable decreases. A linear correlation follows a straight line, while a curvilinear correlation is any relationship that does not follow a straight line. These terms can be combined such that the correlation is positive and linear, negative and linear, positive and curvilinear, or negative and curvilinear. In addition, the strength of the relationship is calculated with a correlation coefficient that ranges between -1 and +1. A negative correlation coefficient is related to a negative correlation between the variables, and a positive correlation coefficient to a positive correlation between the variables. The closer the correlation coefficient is to 1, either -1 or +1, the stronger or more statistically significant the relationship between the variables. Let's look back at the hummingbird experiment. What type of correlation is seen in the hummingbird experiment? This correlation is negative and linear.
Okay, so now we get to do this on our own. So this is looking at a graph of body mass of mammals and changes with latitude. So looking at this, what do we think we've got going on here? Well, it appears that as the body mass is going up, the latitude also continues to go up. So that lets us know that it is positive. And then our next two choices would be, is it linear or curvilinear? And it doesn't look like we have any big scoops or even minor scoops in the data set. So that's going to make it positive and linear. And this is a relationship that you see. If you've ever, I will show you in physiology a picture of a key deer, but the key deer in Florida are smaller than my Doberman. Whereas deer living in Wisconsin and Michigan can like destroy your car. My sister almost had her car totaled by a deer one day. So as you move up with latitudes, you get larger organisms because remember, it helps you to stay warm. So a correlation between two variables implies what? That one variable affects the other. Variables have a linear relationship, not always, it could be curvilinear. One of the variables is the independent variable and the other is dependent. Again, that's kind of something that you assign, and right now we don't really know that anyway. And that one variable may or may not directly affect the other variable. Because a correlation is simply a plot of data, we're not calculating anything. We can't say a cause, we can't put causation on it. So anytime you have a correlation, all you can really say is that, wow, that looks interesting. And that one variable may or may not directly affect the other variable. We don't know. For example, ice cream and drowning. Okay, there's a correlation there with ice cream sales and drowning, but I'm pretty sure most people who drown don't choke on ice cream. There's something else going on there. We use correlations a lot to try to figure out where we should look. Is it worth running a statistical test on it? Are these variables even worth it? You can look at the correlation and be like, all right, maybe I want to spend some of my valuable time investigating these and trying to assign dependent, independent variables and all that kind of stuff. It's fast.
Correlation is a powerful statistical tool used to describe the strength and type of relationship between two variables. But it's important to remember that just because two variables are strongly correlated does not mean that one variable directly affects the other variable.
Thank you. And in the case of the ice cream and drownings, it really didn't have anything to do with the ice cream itself. But a lot of times you have more ice cream sales when it's warmer because more people are swimming. And when more people are in the water, it's the potential for drowning is higher. So here we go. We have the number of predators on one side and activity level on the other. So go ahead and make a quick little graph of the data. And we'll get you guys, we'll take a guess here. Is it a negative or positive relationship? And is it linear or curvilinear? Go. I'll give you guys 2 or 3 minutes to do this. This is a really good exercise to just make a graph on the fly because you never know. Sometimes you'll be out in the field, you're not going to have your computer with you. Your cell phone is like not loving you anymore. So I tend to do these in pencil because you never know. All right. They promised me they're going to buy me a better stylus. I'll get this one. That's great. Oh, that's terrible. That's highlighting. So sometimes when I'm making these, I just think of it in scale. So it's like 40, well, 20 is half of 40, so what's about halfway down the line? It's about there. Yeah, the next one's 10, that's about halfway down the line, that's there. And then we have 5, and then we have 2. Or is it 3? Whatever, it's good enough for what I'm doing here. And then we have 1, 2, 3, 4. So we start here, and come on to here. And I always talk to myself. You probably already know this. All right. You can all be thankful that I don't draw on the board very often. So looking at this, it can be a little bit touchy, a little bit sketchy. Is this a line or is it curvy linear, right? It's a little bit of a judgment call. But yes, I agree, Martin, I would say negative and curvy linear. And I know that's the answer because I looked it up. I didn't just like say that to you guys. But sometimes when you're making your graph, how the data, if it's linear or curvy linear, sort of depends on your perspective on it. So you always want to look very closely. Now in this one, if this were going to be an actual linear graph, I would expect there to be a few data points like out on the other side like this, right? So that would give you like, okay, there's a few outliers down here. Maybe, I don't know, they effed up something that day in the experiment. But because this was so clean, it's negative and curvy linear. So good job, guys. And then I was learning how to use the eraser. I'm so proud of myself. I gotta fix that. I'm working out. If I used to be a system administrator for Unix, I can totally make this iPad work. That's not gonna be a problem. Okay.
You have a question in the chat.
Yes, so how do we know which variable? So here's the great thing about correlation. It doesn't matter, right? You don't know which is the dependent and which is the independent variable at that time. So it's up to you. It doesn't matter what you put X or Y, because in this case, you don't have a strict dependent or independent variable. You don't even know if there's a relationship between it. And so whether it's negative or positive is going to depend on how you set up your data. And there's nothing wrong with that. It doesn't change the relationship between the two variables. Your thought process will be the same. So it's fine.
So I could get positive curvilinear if I had it backwards.
If you had it backwards, yes.
But that's still correct.
If I asked you to do it on an exam, yeah.
Okay.
If I see you graph it, yes, it's absolutely fine. That's the advantage of actually when you write it out and you put the graph together, then I see what you did in your interpretation. It's a lot better than a multiple choice question. A lot of people love their multiple choice questions. I like written questions because then I can see what you were doing and I'm like, oh, well, from that point of view, or, oh, I see they just forgot that negative sign. So I'm only going to take off points for a calculation, not your explanation, because your explanation is fine, right? So however you set it up, if I asked you to do a correlation like that, I would accept it either way because I didn't tell you. They kind of put it on there because they know what the experiment is in the backside. But yeah. And that's also why I made sure to look it up. I'm not above cheating to help you guys. It's not really cheating. We're doing this together. It's guided learning. And because you can ask questions, right? Which makes it a little bit easier. So here we're looking at number of parrot chicks versus number of predators. So poor little parrot chicks. Parrots they have a really good beak, but they're not always best at defending. So describe the strength of the relationship between the number of chicks. So that's our goal here. So let's look at this. Looks like we got a negative curvilinear for sure, but the data are kind of all over the place. I think one of my advisors for my dissertation, he used to say, like, I think if I tilt my head and squint my eyes, I can see that correlation coefficient in there, right? The correlation coefficient is minus 0.1. We haven't done a regression analysis to see if this is even significant, but basically what that tells us is how much does one variable impact the other? Well, yeah, not so much. Really strong would be 1 or minus 1, and we have 0.1. So if I was your advisor and you said, this is totally what I want to do, I'm like, dude, that's a lot of work, and there's not a really good relationship there between that one. So we have, going forward, so that one is pretty negative, and it's not very strong. Is there evidence that the number of predators affects the number of parrot chicks? Very, very, very weak evidence. Again, am I going to give you a $100,000 grant to go to South America, to live in the Amazon and study this? Signs point to no, right? There's a line there. It's negative and curvilinear, but that coefficient is here at 0.1. So it depends. So yeah, you're not going to get some money to do that. Now, the other thing that we can't say that's worth mentioning here is that because this isn't a regression analysis, we're not actually seeing if there's significance here. We can't even put the causation on it. Is there strong evidence? There's not strong. And because this is just a correlation, it's not really even that. It's not even evidence, it's just like an idea, and all these data are scattered elsewhere. And we'll get to it when we get to the regression analysis, but you can have a regression that's statistically significant, but it only explains like maybe 5% of the data. And again, if you're researching, if you were going to be looking at biological compounds that could be cancer drugs, if the relationship is really weak, it might be significant, but again, no one's going to give you money to fund that because that relationship is so weak. It might not be the most important factor that you're looking for. All right, so I think that we're done. But there's, when you're doing your research, there's nothing wrong with starting with the correlational analysis just to see what's going on. There can always be a third factor, like a parent competition that we'll talk about later, but that's trying to figure it out. Before you run a regression analysis, you're going to want to put it in the order you think that this causes this, right? Number of chicks related to predators. But if you're just throwing the data out there, that's no big deal. So, all right, gang, thank you so much for your time and attention. I hope you have a great weekend. Everyone stay safe. Wear your masks. I just looked, the infection rate is down to like 2.6% in Pinellas County yesterday. Really, really low. So it's making a difference. And we can all get back to our crazy Florida lifestyles, whatever that is for you. And Ricky's like, I'm a dad. I don't have a crazy lifestyle. But all right, guys. Love you all. Take care. Bye-bye.
Yeah.
See ya.